So yesterday night I was preparing the talk and I asked to myself why I'm here. And I think that I didn't solve the question and I don't know what I'm going to tell you. So it will be a surprise for both of us. So I decided to put few slides and tell you where we come from and why along the last 15 years we moved from one technology to another. So we heard about personalized medicine. I think it's a very complicated issue. I think we are just simply on the tip of the iceberg. Detection of targets is not enough. There is an editorial in Cell this morning, I just looked at before I came here, where it's clearly evident that there are pathways, they synergize, they swing, they move along therapy, so it's a mess. So we have to be very, very precise in what we are saying, what we are doing. The identification of a target is not enough. It's not enough because it's a simple target definition. We need to find out in a functional way how it works and how the equilibrium is mismatched based on therapeutic intervention. I think it's much more complicated than that. So a long time ago, when I was an assistant professor at Columbia, I decided to pick T-cell tumors for two reasons. I had to survive with my R01, and the competition is ferocious, and therefore I picked a very orphan disease. So these are very orphan disease by definition, and I was not happy enough, and I picked another super orphan disease that is, I don't know. Here, I picked this one, anaplastic large cell lymphoma kinase, positive tumors. This is a very, very orphan disease. When we start to work on this, no one really worked, and we made a little niche. And when ALK was discovered in lung cancer, our competition became ferocious. So these tumors are rare, they are very heterogeneous, but we don't know anything about the pathogenesis of these tumors, with rare exception. And the exception is anaplastic large cell lymphoma kinase. So the presence of a fusion that links an anaplastic lymphoma kinase in a very ectopic content and drives transformation of these cells. Survival is poor, very poor. We have no therapy. We are using the therapy that we have used for years in B-cell lymphoma very unsuccessfully in T. So largely are CHOP-based protocols, and they are very unsuccessful. There is a lot of mess regarding the relationship between different kinds of tumors. We really don't know where they arrive, how they move from one entity to another, if this phenotype is driven by genetic disorders. So there is a lot of chit-chat among pathologists, but we have no really solid data. So why we need to move forward and integrate technologies? Because we don't have really models. We are really blind on this, and so we need to generate efficient system to analyze this particular context. And in reality, we have few cell line and few mouse modeling. So I think that from there we have to pick other alternatives. Two words on the main gene that is responsible for this subset was discovered by Steve Morris and Tom Luke a long time ago. Now it's becoming very popular, so everyone knows about ALK, so I'm not going to talk about. But basically, there are two major, let's say, molecular events that are critical. We have a dimerization domain, a 5' end, and we have a a catalytic domain that is of ALK that is driven by the heterodimerization or homodimerization. So we have signaling, constitutively signal. There are plenty of genes that they are fused to ALK. I mentioned before that they were mainly found in ALCL, so in T-cell lymphoma, later on in B, in some mesenchymal tumor, and more importantly in a lot of solid tumor. Basically, every kind of solid tumor that we tested has ALK kinase fusion in different percentages, but around 2 to 5%, and we have colon, we have lung, we have liver, we have prostate, we have ovary, renal, and so on and so on. So it's a very pharmaceutical targetable molecule in this moment. So we proved a long time ago that was oncogenic in mouse modeling, and the key transformation of this is related to ALK signaling, but more importantly to STAT signaling. STAT in this context is absolutely required. But 2 words on personalized medicine. This is a fantastic oncoantigen. ALK is not generally expressed in normal cells, so it's a real oncoantigen, could drive immune responses. They are present in patients with this disease, but not efficient to basically stop the process. A long time ago, we designed a gut shot approach, DNA-based approach, and we proved a long time ago that the targeting of this molecule is efficient to stop disease in animals and to regress disease, and in combination with chemotherapy is very efficacious. But in reality, mice are mice and not human, and therefore the immunological response that we have in mice cannot be simply translated in human. And therefore, we now have a new model where we have reconstituted energy mice, so highly immunocompetent animals, with CD34 human cells. We have a full repertoire in BNT, and now we are vaccinating these animals with different peptides. So this is the approach that potentially could be applied for ALK-positive tumor, highly personalized. But to do a personalized approach, we need to find out who are the lesions that they drive these tumors, and as many others, we use the same technologies. We started a long time ago with an high-throughput proteomic mapping approach, and for example, we discovered the ALK, but other kinases, they phosphorylate ATIC, and therefore they dampen the efficacy of methotrexate. So this is a simple approach that should drive at least scheduling in different diseases. Now, obviously, as everyone else, we are doing whole genome to find new mutation. The strategy is relatively simple. So we are applying old techniques, so gene expression profiling with specific algorithm, alternately RNA-seq or exome sequence. I will present few data on that, very, very, very short. So we focus on ALK minus with the prediction that if there is a subgroup that is driven by a kinase, maybe there is a second group that is driven by kinase, but we didn't discover yet. And the response is yes. So we have found 15 new translocations, some of which they bear kinases. A more interesting one, some of which they have a real chimera. In other words, the 5' end that is dimerizing the kinase is a transcription factor and is fully oncogenic. So now we have 2 hits in a single molecule. And these are repeated hits that they appear in different populations. The frequency is relatively low, probably, around 2-5%. But if we put all together, we now cover over 80% of the ALK minus as kinase-driven processes. I will talk about personalizing Tumorgraft because it's becoming my obsession. So everyone that listens to me along my talks knows that it is becoming my obsession. So the idea is relatively simple. We heard in the first presentation that many of the drugs, they don't work. So that means that probably we don't have a right pipeline to define efficient drugs, and we are missing probably a lot of good drugs along the way. So this is an old review that came up in an old paper, but bottom line, the message is we need to have better representation, and probably this is a pretty good model that we can apply to. So these are mice developed by many investigators that now are put on the market by Jackson Laboratory that we have a collaboration with. These are very unique animals that they, in addition to the classical BNT dampering, they have also a knockout for the gamma receptor. Therefore, they don't respond to a lot of lymphokines. So these are really empty bags that they don't respond to immune signal. And these models have been now used for generating alternative tumor graft, either in node-skid classical or more recently in node gamma 2 minus, where we now implant different kinds of diseases. I will present my experience, so the experience of our group. So obviously, these are not perfect, and they look like, but they are not primary tumor. And I think that a lot of publications that came up recently, they missed this point, and they jump to conclusion that they are not really solid. So we need to be cautious. So we devised a strategy in which we are now characterizing serial tumor graft, and we are doing extensive molecular studies along the way. And these are basically some of our tumor grafts that are implanted in many animals. They are frozen down, they are viable, we can re-implanted, and every new tumor is expanded, and every tumor is now analyzed by gene expression, RNA-seq, SNP array, and exon sequence. We need a lot of grants. But bottom line, we can implant it successfully in more than 80% of these rare tumors, and the curve along the time is basically very similar if we consider multiple implants, or if we take frozen viable material from another institution and we implanted and we grow the tumor. So we establish libraries. And these libraries, they look all alike, not only as a growth, but also as physical characteristic. These are unique model. This is an anaplastic large cell lymphoma implanted subacute. It's a very large tumor, but this creature is capable to chit-chat with all the lymphoid tissue of the mouse that are totally empty. They go in the lymph node, they establish the right connection, they home in the right environment, they display the right configuration, and eventually they disseminate. So something that we never see in cell line. This is quite unique behavior. They become metastatic, they invade very different organs, and we can trace them with specific marker. In this case is tumor necrosis factor member gene, so it's a CD30, we have anti-ALK and so on. We can trace down all this. How they go there? They go by blood, they invade the blood, we can pull out the cell population, the tumor cell population, even with small masses along the way, and they colonize. Their phenotype is maintained along the way. But more importantly, we can now start to understand if they are different. And for example, this is a simple experiment, so we have now a series of tumor grafts that when are analyzed by gene expression in unsupervised analysis, the primary tumor goes with the tumor graft. It's not a surprise. For me, it's a surprise because in lymphoma, the host component is very well represented, and in the mouse tumor graft, we have basically tumor cells. So that means that the signature of the tumor is so strong that it links together primary with the tumor despite the normal cells. Now, if we do classifier and we now analyze how this classifier, they emit and they recognize a primary tumor set of 249 anaplastic large cell lymphoma, each one of them recognizes a specific subset of individuals, and these individuals, they have a very unique clinical behavior. So simply based on the classifier, we can now stratify clinical behavior among untapped population. A lot of the signature, as I mentioned before, is driven by the milieu, so the host. So this is a gene expression profile that compares the signal of the primary tumor versus the tumor graft, and all these genes are lost. These genes, they have a critical impact. They allow the interaction host-human. They are lymphokine-driven signaling pathway. They are uniquely expressed in primary, absence in tumor graft, and the mouse is now providing similar signaling, and therefore now we know who belongs to and how the host is now supporting the tumor. If we do SNP array, we can identify lesions that are present in primary and tumor graft. We can see changes, and some of those they swing and some they remain, and therefore we can target it. And I give you a few examples. In this case, we found, for example, that this particular tumor bears by RNA-seq a new translocation that breaks down NF1, so removes the inhibitory signal of RAS, and therefore this tumor has a fusion that is present in the primary and the tumor graft. The NF1 gene is broken down in one allele, and on the second allele bears this translocation that basically inactivates the function NF1, is as a high signaling pathway, and when we use AZD6244, we have a response in the mouse. Therefore, this RAS pathway is important in this particular tumor graft. But these tumors, they very well recapitulate, for example, response to chemotherapy. This is a patient that was responsive to CHOP. The tumor graft treated with CHOP is responding, is relapsing. These tumors are implanted again, they respond and they relapse. So it's following exactly what's going on in the patient. The patient went through 4 cycles of CHOP, she relapsed, went again, and so on. So it looks like that we are recapitulating quite well that. And now if we use an alternative strategy, we can save the mouse and therefore potentially the patient. This is an ALK positive tumor. This is a kinase inhibitor developed by Cephalon. We have been working with them. Now they disappear, but for a long time. This is a highly selective inhibitor of ALK. All the mice that are treated with this particular compound are alive more than 18 months from treatment. As you know, these tumors, they have CD30, and this is a very hot molecule. It's called SGN35. It's an antibody against CD30 that is linked to a toxin, and is probably the new compound for a refractory patient on ALCL. And here we have been using in these animals and proving that there is a relationship between tumor mass and drug. So bigger is the mass and less is the response. But more interesting that if now we combine classical protocols in a tumor that was always relapsing, now we combine CHOP and anti-CD30, we have total response in these animals. So these animals are free of disease. So combinational therapy based on target, I think, is a pretty good example. Now the last example that I finish, this is a patient, rare patient, with a leukemic stage of anaplastic large cell lymphoma. This patient, they died literally in weeks when they develop leukemia. So this is basically her poor course. She was diagnosed in December 2011, and in February she became leukemic, and soon after she died. So very rampant disease. Something that if we look the chart overall survival in the literature, it looks like that this does not happen, but this happens. So we approach this particular tumor using multiple pathways, RNA-seq, and we discover a new translocation between TRAF1 and ALK, never discovered before. TRAF1 is an adapter of TNF signaling pathway, and this particular molecule now is bringing to the fusion a domain and a cold domain, TRAF domain, a cold domain, homodimerizing, and also binds to TRAF2. So now is engaging preferentially an NF-kappa B signaling pathway, in addition to classical ALK. But more interesting, we found that these animals, they become leukemic when we implanted these tumors. And if we treat it with the ALK inhibitor, we have a pretty good response, but we don't cure these animals. These are resistant to ALK inhibitors. And why? Probably for two reasons. They acquire a p53 loss, and now they have a c-MYC amplification. And the c-MYC amplification can be now dampened by a specific inhibitor that is called JQ1. This is a bromodomain inhibitor that blocks MYC. So the combination now of a MYC plus ALK makes the tumor more sensitive to therapy. And we have developed tools to do this particular approach. This is cute if it works, and I will show you in a second that we have now designed a software that they allowed us to analyze the animals when they are treated. This is an MRI, and now we are reconstructing, for example, the presence of, you don't see it very well, but here there is a liver, there is a spleen, and the kidney. These are animals followed every 3 days by MRI, and we reconstruct the MRI, and we determine the volume, and now we are capable to determine oxygenation. We can determine pyruvates, for example, concentration, glucose, and so on. So we have now readouts that they allowed us to have very different and very sophisticated models. I think that the people that worked on this project are many. We had a framework 6, but not 7. We are working on it. We have put through many applications. Probably we are making you very busy. And there are plenty of people, and the collaboration are all over the place. I want to simply mention the European T-cell lymphoma study group. When you deal with orphan disease, you need a lot of friends. Otherwise, you are not capable to make any contribution. And this is probably the reason why I'm here. Thank you.